Assalamu alaikum wa rahmatullahi wa barakatuh. This is Professor Alaa Eissa, the chairman of the Department of Aquatic Animal Medicine and Management, Faculty of Veterinary Medicine, Cairo University, a veterinary consultant for the Suez Canal fish farming company, or the Mecca fish mill company, as well as a consultant for the Italian Egyptian marine development project. Dr. Muhammad Fathi Osman, my dearest professor and teacher, who I have learned a lot from his knowledge and politeness and humbleness. He is really a great man. Thanks for the invitation, and I hope you like my lecture. Thank you all for attending this session, and with the lecture titled Fish of Stress-Resistant Diets and Other Special Diets. First of all, everyone knows that the disease in aquatic environment usually results from an interaction between the host and pathogen and the environment. Three main circles intermingling together. These three circles represents the major components of the aquatic environment, either natural or introduced. So the host, the first cycle or the first circle, represent the host, where any compromisation or any impact on the fish to their immune system, the pathogens which is living in a very natural way in the aquatic environment or very ubiquitous in the environment, they will surely host, and as a result a disease will result. Also, the environment, including both of them, the pathogen is the major player in this issue, where any deterioration of this environment, either through impact on the salinity or impact on the oxygen content, dissolved oxygen content, temperature, or maybe dissolved solids, all of these components or limiting factors are a key player for induction of disease when they get worsened. So the disease is a triangle that is resulted from an equation between host, pathogen, and bad environment. Pathogen may increase in number or maybe may increase in virulence, depending on the deterioration of the environment, where some factors in the environment could result in enhancing the multiplication of this pathogen and further increase their virulence in a very drastic way. Another aspect, the bad environment and deteriorated environment will, of course, impact the host immune system and, of course, will destroy their barriers or resistance against pathogen invasion. These three circles is called UNESCO cycle, disease dynamic cycle, and everyone in the fish health research aspect should know these three triangles. The disease cycle in general is conformed of or consists of host, environment, and pathogen. And the host, probably this kind of major circle, is determined by the age of the host, the sex of the host, the species, the strain, the individual variations, anatomy of the host, feeding habits, predation, spawning. Also the environment, which is containing both pathogen and the host. They have limiting factors or determinant factors, which are the salinity, where some salinity percentages would allow certain pathogens to invade fish and others will limit their growth. As we all know, the freshwater fishes has a 5 up to 8.5 as a pH, while the marine environment a little bit increase than this aspect to the level of 9 maybe. Any increase than this level or decrease than this level will reflect with a disease. Some diseases like columnaris disease were favored with the alkaline pH, and some diseases were favored with the acidic pH. Dissolved oxygen is a very critical and limiting factor for any kind of creature living on planet Earth, including the aquatic animals. As you all know, there is a limiting factor which limit the life in aquatic environment when it goes back, when oxygen goes back or goes down to 2 ppm, and sometimes it goes to 1 ppm, which means there is no life. However, the optimal level could be 5 to 6 ppm, and usually this number changes from the early morning up to the noon and maybe to the sunset, depending on the temperature of the surrounding environment. The peak of the oxygen tension is at noon or maybe in the afternoon, and the low is very low level of oxygen is the early morning. Also, the carbon dioxide level is a natural environment. It is very determinant factor in ocean, seas, and so on, and it plays a major role in the process of global warming and melting glaciers, as well as the sea level rise and the ocean acidification. However, in cultured environment, it has a little bit lowest impact. Ammonia is an extrinsic factor that introduced by bad management and intensification, as well as bad quality feed stubs, as well as overfeeding, as well as increase the frequencies of feeding, as well as high alkaline environment, as well as sewage, as well as agricultural drainage water. Hydrogen sulfide, turbidity, and sewage and agricultural drainage are impacting components of the aquatic environment. When you see a deteriorated environment, aquatic environment, it means that the left side column will be jeopardized and their immune system will be damaged, while the other right side, which is the pathogen, will be enhanced, increased in number, increased in virulence. Whatever it is, gram-positive bacteria, gram-negative bacteria, septicemic bacteria, rickettsia, viruses, dermatophic fungi, systemic fungi, parasites, toxic dinoflagellates, and so on. Now we should know that the main problems facing aquaculture are feeding problems, water quality problems, stocking density problems, disease issues. Thus, when you know these four main issues, you know why you are formulating a special diets, which is stress resistant diets, and maybe other formulations that are facing this kind of troubles. Let's go through the feeding problems. The timing of feeding is a determinant factor for occurring of a problem in a fish farm. Thus, we should know that the optimal timing for feeding should be enough time after the sunrise or at the mid-noon, while the erratic one is at the dawn or maybe at the early morning. Also, the frequency of feeding should not be only once to 2 times or maybe 3 times per day. The amount is temperature dependent, and the farmer should be very malleable with the temperature while offering the feeds or the pellets to the fish. For example, we can feed with the maximum potency or maybe the maximum potency of feeding at 25 degree maybe to 28 degree, while after 30 to 31 or 32 you should minimize your food. At 35 or maybe 33 you can stop feeding it. Also, distribution of the feeds is very crucial for obtaining good sized fishes, and if we got any distribution of the fish pellets, then you will get a problem. So distributing the locations from 3 to 6 locations on both sides of the fish pond is very optimal. The quality of feeding also is very important. Go for high quality feeds from known or authenticated fish feeds companies to ensure good feed conversion rate, to ensure good quality fish mussels, and to secure the consumers, the human consumer. How can you differentiate between high quality and bad quality fish feeds? Number one, fish feeds should contain high quality fish meal that contains pure essential amino acids, methionine, lysine, tryptophan, and so on. Fish feeds contain high quality oils. Fish feeds should be mycotoxin free. Added values which are talking about in this lecture is the special diets or special stress resistant diets, those including anti-mycotoxins or mycotoxin binder agents, or maybe long-acting vitamin C, or maybe probiotics, prebiotics, immunostimulants, intestinal habitat modulators, enzymes, vitamins, maybe phospholipids, and so on, hormones, and so on. Types of feeds are very crucial also, because as you know, every stage in the fish cycle have their suitable fish pellets or maybe fish feeds. For example, the very tiny young fishes or juveniles should be offered the powdered diet, while those a little bit bigger than juvenile stages should get 1 to 2 millimeters, 2.5 millimeters, 3 to 3.5 millimeters, 4 millimeters until the end of the cycle meters. Also, from you can decide either you have to offer a floating or sinking fish pellets. However, in some fishes like tilapia, at the beginning we can offer sinking pellets, then we can shift it to floating low protein pellets, for example 25% protein pellets, to minimize ammonia and to minimize this kind of diseases originating from excessive protein offering during the hot summer. Also, there are some fishes in the marine aspect that have been only offered trash fishes, dead poultry carcasses, and so on. So the possibility of disease spread is high, problems is high, and other problems. Also, using poultry manure in the process of feeding of fish is a very bad idea, especially if you know that many of the fish diseases are coming from the poultry origin. The main aquaculture and problem may be methods, which I call it methods because it's really methods. High stocking density overfeeding violated biosecurity, and unfortunately most of the natural fish ponds in Egypt, this kind of semi-intensive earthen fish ponds, either in the freshwater or maybe marine aquaculture, either in Kafr Sheikh, maybe in Sharqiya, or maybe in Fayoum, maybe in Damietta. All of them are rearing large animals, maybe in the vicinity of the fish pond or maybe a little bit closer to them. But for fish, they are commensal for these animals, but for fish they are highly pathogenic. For example, Streptococcus and Enterococcus and Euromonas and Yersinia and Edwardiella and so on. Poor water quality is very detrimental and crush in inducing diseases and the problems within the fish farm. These are some pictures for the companion animals and the large animals and the poultry that are living in a very peaceful way with a human at this fish farm, however in very crusher and drastic way to fish. So this is should be forbidden. Here is the story of the large animals, which is a kind of source of infection for fish. So we can conclude that the messes and the problem are also unhygienic or non-disposal of dead fishes, where some of the fish farmers are always leaving the dead fishes within the fish pond, and they are claiming that these dead fishes are giving an immunity to the live fishes, which is a myth, and which is a kind of erratic conclusions, should be corrected and rectified. Also, minimizing water columns is another myth that is established by the natural farmers, which is very bad idea because they are concentrating toxic materials as well as pathogens. Also, there is a big issue here in the Egyptian market, which is using adulterated or unregistered drugs for treating fish diseases. And we need to regulate this through the veterinary and veterinary associations and maybe Ministry of Health and Ministry of Agriculture. The picture in front of us is showing dead tilapia fishes. They are attended within the fish pond without collection, and this is one of the major violation of the fish pond biosecurity in the Kafr Sheikh province, where the Baladi system or the native rearing system and some of the methods and erratic management are prevailing over there. Also, some of the dead fishes with lesions of motile Eromonas septicemia with severe ulcers and even the muscles are exposed were left unattended on the bridges between fish ponds on the soil, and these fishes are left to be eaten with the aquatic birds, maybe herons or maybe this kind of ducks and so on, and could be eaten also by dogs and cats, which represents a big and major violation for biosecurity standards within these farms, because most of the birds who have eaten these dead infected fishes, they fly on the pond and sometimes they drop their feces with a continuation of the disease cycle, and we became in the middle of an endless way of disease termination. This is a bridge between two fish ponds, or the bank between fish ponds, were showing the accumulation of dead fishes over the soil, which represent also kind of major violation to the fish biosecurity, fish pond biosecurity, and another source of infection and dissemination of disease within the pond. So how can you control such problems who are totally convinced of not collecting dead fishes either from the fish pond or from the bridges? Corrective action. Usually to control such problems which we have detailed or entailed, we have to have what is called a corrective action. Number one, we have to enhance the water dynamics by improving irrigation, drainage, enhance oxygen formation in ponds using water bottles or injectors, oxygen injectors, or water recycling system. Also, we have to adjust stocking densities to the pond area and the water column. Also, we have to manage the feeding regime to be adjustable or changeable with the daily water temperature, especially at the hot summer, to control the ammonia release and to control the nitrogenous compound waste within the water and to control the accumulation of organic matter within the fish pond. This is a picture of some of the water bottles and some oxygen injectors and some of the fountains made within some fish ponds to enhance the water dynamic. Number 4, within the corrective drainage and the drying disinfection of the fish pond bottom before or between the rearing cycles to avoid the accumulation of the pathogens from the previous cycle and to minimize the amount of organic matter accumulating at the bottom of the fish pond. We can use calcium oxide or maybe calcium hypochlorite or maybe chlorine or maybe any other potent disinfectant to control such a problem. Also, we have to disinfect nets, feeding tools, utensils, and inanimate objects before using in another pond or between one pond to another. Also, we have to use balanced or improved fish feeds or containing anti-stress or anti-mycotoxin or immune stimulant and so on for controlling the problems occurring due to fish feeding during the fish production cycle. And these feeds should be from authenticated fish feeds companies to be sure from the quality of such fish feed products. We have to adopt a strict biosecurity strategy that will be the key step for clean rearing cycle. And this can be obtained through vaccination, if possible, optimal disinfection, prevent entrance of invasive species and unwelcome creatures and large animals, aquatic birds, pets, rodents, amphibians, and so on. 5. Number 8, using registered authenticated drugs including antibiotics, disinfectants, probiotics, yucca, hydrogen peroxide, acidifiers, vitamin C use, and so on. Also, we have to call for an aquatic veterinarian for help in case of abnormal finding and disease issues and mass mortalities. Don't stay at home or don't stay reluctant in front of mass mortalities or diseases because it prevails and the losses become enormous. So now we have two rising questions about the sustainability of tilapia aquaculture in Egypt, which is number 1, can tilapia resist the continuous waves of mass mortalities worldwide among all life stages? Now we have viruses which we have never heard about before. They are hitting tilapias very strongly. We have a lot of emerging bacteria, a lot of problems that we have stuck during the production cycle because of the changing immunity and the resistance of tilapia nowadays. Also, the second question is, can tilapia resist the increasing number of emerging diseases affecting tilapia worldwide among all life stages? Now,
So we have a possible hypothesis growing risk. Number one, the extreme climate changes, which is high environmental temperature, partial pressure, humidity, dissolved oxygen, carbon dioxide levels, all of these issues are impacting the Nile tilapia's growth. Treatments during the mono 6 production, most of these formulas are poorly stored, inefficiently mixed, and dosage is miscalculated. Number three, emergence of novel killer viruses and bacteria specifically targeting tilapia. Number three, the rising aquatic pollution associated with erratic usage of agricultural drainage water and sewage. Continuous exposure of fish to pollutants will act as endocrine disruptor, which consequently will act on the growth and the sustainability of the fish. Overstocking densities of fish within confined areas with short water columns leads to increased risk of disease spread and eruption of new disease by cohabitation. Number five, extreme shortening of rearing cycle by intentional overfeeding is one of the major problems too. Genetic manipulation in favor of broodstocks or male and super male production could be another dilemma, because we don't know if some of these genes which have been played with or may be manipulated, if it is linked with another bad gene or not. For example, if the enhanced growth gene is linked to disease issue or not. So it needs further studies.
The second issue is the probiotic. What are the probiotics? Probiotics are live microorganisms when supplied in adequate amount to cultured organisms, confer health benefits of the host and sometimes surrounding aquatic environments. The probiotics are live, non-pathogenic and non-toxic microorganisms without undesirable side effects when administered to the aquatic organism. So, probiotics are live microbial feed supplements, which beneficially affect the host animal by improving its intestinal balance. We can say that the range of probiotics examined for use in aquaculture has included both gram-negative bacteria and gram-positive bacteria, also euphrags and unicellular algae. How probiotics work? Everyone knows that probiotics have antiviral activities against infections. They produce inhibitory chemicals. They improve the water quality by participating in turnover of the organic nutrients accumulating at the bottom of the fish ponds, and also toxic nutrients, toxic ammonia and amines in aquaculture system. They enhance immune response by increasing the phagocytic activity or the cellular activity of leukocytes. They compete for nutrient otherwise consumed by pathogenic microbes like Photobacterium, like Aeromonas, like Strep, and so on. They also compete for adhesion site and food with the pathogen surface and prevent colonization of the organism on the villous surface. Probiotics are source of nutrients and secretes variety of enzymes to increase feed degradation, assimilation, enhancing its nutritional values. So probiotics are...
Okay, the picture in front of us is explaining the role of the probiotic, either the dietary probiotic where it works directly on the intestinal villi of the fish. There are other topical probiotics which work on the fish skin, and a part of it work on the water of the pond, minimizing the ammonia, minimizing the hydrogen sulfide and other organic matters. These together could be a backbone of a bioremediation process for the deteriorated aquatic environment. So we are working on the digestion of food and the immune response of the fish, and we are working on improving of the water quality of the fish pond, and together we are working on a bigger picture, which is fish good health of product, and in the same time we are minimizing disease spread, minimizing the risk of being endemic, of a disease to be endemic in a fish pond, and so on. So this is the picture in front of us.
Great. Then what are the prebiotics? They are a little bit different from the probiotics. However, they are part of the natural structure of the probiotics, but it's non-live part. A prebiotic is defined as a non-digestive food ingredients that beneficially affect the host by selectively stimulating the growth and or the activity of one or limited number of bacteria in the colon. Thus, they improve the host health. How? For a food ingredient to be classified as a prebiotic, it should be, number one, neither hydrolyzed nor adsorbed in the upper part of the gastrointestinal tract. Number two, be fermentable by intestinal microbiota. Number three, be selective substrate for one or limited number of beneficial bacteria to the colon, which are stimulated to grow and or are metabolically activated. Number four, consequently be able to alter the colonic flora in favor of a healthier compost. Also great. So prebiotic is another great thing. Prebiotic brings about a specific modulation of the gut microbiota, particularly increased numbers of bifidobacteria, or maybe lactobacilli cell count, or decrease in potential harmful bacteria. That is sufficient criterion for health promotion. The most common prebiotic used in fish are carbohydrates like inulin, fructo-oligosaccharides, short-chain fructo-oligosaccharides, oligofructose, mannano-oligosaccharides, trans-galacto-oligosaccharides, which are non-digestible but can be fermented by the intestinal flora. Another great thing, which are the real scream in aquaculture industry nowadays. What are the organic acids? Organic acids are organic carboxylic compounds. What does it mean, organic carboxylic compounds? They are carboxylic compounds of general structure formula RCOOH, whose acidity is associated with their carboxyl group COOH. They are weak acids because they partially water to form a hydrogen ion and a carboxylate ion, for example acetic acid, or maybe peracetic acid, or maybe formic acid, or maybe propionic acid, and so on. Dietary acidification by addition of organic acids has been widely used in animal. Organic acids have become a promising feed additives to improve gut health and performance in most of animals, especially aquatic animals like fish. How organic acids work, or maybe the mode of action or mechanism of action, whatever you call it. The pH decreasing action of organic acids in the stomach and the small intestine contributes to an improved activity of digestive enzyme. Number two, some organic acids can penetrate the cell wall of certain types of bacteria, disrupting the normal physiology and inhibit the growth of pathogenic bacteria. Number three, organic acids also used in fish feeds to reduce the potential threat of microbial contamination, including pathogenic bacteria and molds or fungi due to aspergillus or maybe penicillium or maybe fusarium that may grow during feed storage. This picture shows the potential of organic acids and how in diets, pH and buffering capacity, harmful bacteria intake, feed working on all of them. So the fish is eating all of that, reaching to the stomach. So stomach now have is full of harmful bacteria, enzymatic activity, mineral solubility, and so on. Then the fish is releasing in feces their phosphorus load, microbiota load, risk of cross infection, and deteriorating the water quality. And the intestine, epithelial cell proliferation, nutrient availability, mineral availability, beneficial microbiota, colonization of harmful bacteria, gut health is impacted. So the antibacterial mechanism of action of organic acids in animal cells could be for such problems. Okay, they are colonizing this kind of places where pathogenic bacteria are looking for to sit on. So they separate them from these locations and they are firing them out. Okay, also they have limiting factors by modulating the pH of the intestine and so on, not allowing this kind of bacteria to grow in a very favorable condition. We can say that in the small intestine or the midgut, some free organic acids could work. Stomach and foregut, free organic acids could work. In the large intestine or the hindgut, microencapsulated acids and essential oils are the most suitable. So the most suitable to the small intestine is free organic acid, stomach, but the large intestine, microencapsulated acids and essential oils are a little bit better. The commonly used organic acids. Number one, individuals are a combination of organic acids such as propionic, sorbic, benzoic, butyric, malic, lactic acid, and acetic acid. Also, the salts of these organic acids such as calcium propionate, potassium sorbate, sodium potassium diformate, butyrate, and sodium benzoate are also available at the market. We are not talking about commercial. We are speaking about active principle as a kind of neutrality between the producing companies. Enzymes. Another player in the anti-stress formulas of exogenous enzymes like phytase, carbohydrase, protease, lipase are used in aquaculture feeds to overcome the negative effects of anti-nutritional factors. They also improve the digestion of dietary components and enhance the growth of fish and FCR. How enzyme works? For example, phytase is an enzyme specific to hydrolyze indigestible phytate, has been increasingly used in fish feeds during the past 2 decades. However, it's very, very critical for herbivorous and omnivorous fishes, but it's of no value in case of carnivorous fishes or carnivores. According to Paytel, up to 80% of the phosphorus in plant seeds are in the form of phytate. Thus, the digestibility and availability of phytate phosphorus for fish is very low, consequently lost to the environment as a waste. And we as scientists should work in minimizing this kind of waste to avoid any kind of enhancement of disease issues in the aquaculture. The use of phytase enhances bioavailability of phytate phosphorus. Obstacles facing the use of enzymes in aquaculture. As you all know, effect of temperature and the stability of enzymes applied in the feed processing. Leaching also, loss of the enzymes in water, and effectiveness of some enzymes, for example microbial enzymes that have 37-degree optimum temperature, when applied in cold water. Aquatic animals that have low body temperature are constraints regarding the effectiveness of enzymes application in aquafeeds. So in all means, high temperature or low temperature are impacting and jeopardizing the significance of these enzymes. They are a little bit not widely distributed in the Egyptian market. Hormones. Hormones are various hormones have been used for different reasons in aquaculture. Some of them are used for growth, some for steroid hormones, some for sex development, some for producing monosex tilapias, and so on. For instances, growth hormone and thyroid hormone like thyroxine plays an essential role in the stimulation of somatic growth and survival of tilapia. Monosex fish production is common practice in aquaculture and possible in 47 species using steroid hormone, methyltestosterone, as one of the potential players in production of the male tilapias in the Egyptian market and worldwide. Another great issue in the fish feed industry is the mycotoxins. Does the mycotoxins have Egyptian aquaculture market? I could say yes. Why? Because of the bad storage of the fish feeds in the open environment in fish farms. Further, maybe the bad quality fish feeds ingredients. Okay, so what are the mycotoxins and their impacts on aquaculture? Mycotoxins are toxic metabolites produced by diverse group of mycotoxic fungi. For example, Aspergillus flavus, Aspergillus niger, Aspergillus ocretius, that contaminate agricultural crops prior to harvest or maybe during storage post-harvest. Mycotoxins represent a serious problems in fish production worldwide. Mycotoxins effect includes the reduction of weight gain and feed efficiency, causing liver and kidney damage, worsening the overall health of the fish, and which can result in serious economic losses and implications to the fish farmer.
This is the drastic impacts and the lethal impacts of mycotoxins on different organs of fish. For example, they impact the kidney health. Those ochratoxins and aflatoxin B1 causing necrosis in pale kidneys, decreased resistance to oxidative stress. Those are impacting the fertility, their allowance, causing reduced spawning frequency, change of relative fecundity, acceleration of sexual maturation. Also, the other groups, which is aflatoxin B1 and fumenzin and ochratoxin A, are impacting the GIT or gastrointestinal tract, causing reduced growth and bad weight gain and loss of appetite. Another group, which are the aflatoxin B1, fumenzin, and ochratoxin, they impact the liver, impacting the detoxifying function of the liver, and so on, and consequently have a greater impact on health and on growth. Common complication: suppression, survival rate, and bacterial cell count, teratogenic effects, alteration of SA and SO ratios, increase, and so on.
Most of the fish mycotoxic fungi are in the pre-harvest era or stage, where deoxyvalinol and zearalenone and fumonisin are all bad players in this area. In the post-harvest stage, the ochratoxin is the mycotoxic player at this stage. Post and the pre-harvest mycotoxins are mainly the aflatoxin. So, to alleviate and to prevent and maybe to hinder impacts of the mycotoxin on fish feeds and consequently on fish health, we have to have what is called mycotoxin binders. 0.5% of hydrated sodium, calcium, aluminosilicates, HASCAS, which has been called also HASCAS, could effectively reduce aflatoxin B1 in Oreochromis niloticus and maybe in other fishes. HASCAS binds aflatoxin in the gastrointestinal tract, thereby reducing overall bioavailability to the bloodstream, with consequent good impact on fish health. Another thing, which is 1% bentonite clay, reduce the grass inhibitory effect and bioaccumulation of Nile tilapia by research, as well as tissue lesions due to aflatoxin B1. Due to the importance of the mycotoxin binders, I am showing you this diagram or picture where global feed mycotoxin binders and modifier markets appears here or trend. 2.5 billion US dollars are worth as you call it, losses due to mycotoxins. $2.8 billion are spent to alleviate and to counteract these effects by what is called mycotoxin binders. Another key player in the special immunostimulant is a naturally occurring compound that modulates the immune system by increasing the host's resistance against disease that in most circumstances are caused by pathogen. Example for those immunostimulants may be garlic, may be black seed, may be pine oil, may be thyme oil, or maybe thyme, and so on. Amino acids. Amino acids are very important and the crucial components of the stress diets. Anti-stress diets, stressful husbandry in general condition affect amino acid metabolism in fish, and under some circumstances or stress situations, an increase in the requirement of certain essential amino acids occurs, which is probably related to the synthesis of protein and other compounds related to the stress response. The role of specific amino acids and their metabolites on key metabolic pathways that are necessary for growth, immunity, or resistance to environmental stressors and pathogens have been already reviewed in fish, and they are famous now. Thus, amino acids not only serve as constituents of the protein and energy sources, but also can be converted into important biochemically active substances that is defying or fighting stress in fish. What are those amino acids that are very important in this cycle? Arginine is the precursor for the synthesis of the nitric oxide and polyamines in higher vertebrates. In fish, nitric oxide production plays a major role in cellular defense mechanism against bacteria as well as other pathogens. Moreover, dietary arginine can increase of fish following some innate immune mechanisms and disease resistances, disease resistance with, for example, Photobacterium damselli, or maybe Motyle Aeromonas septicemia, or maybe Streptococcus, and so on. This is just a model of bacteria, pathogenic bacteria. Also, branched chain amino acids, or PCCA, like leucine, isoleucine, and valine, have an important role regulating protein synthesis and skeletal muscles, being leucine the most effective in regulation of this process. An increased proteolysis activity is usually observed in fish under stressful situation, together with a decrease in plasma level of branched or branched chain amino acids. Therefore, dietary supplementation with BCAA, especially leucine, appears to be a crucial or promising tool to mitigate negative effects of stress in fish. Vitamin C. Vitamin C has been the object of first work on stress alleviation through vitamin supplements for decades in both fish and superior animals. Moreover, from a nutritional perspective, the vitamin C content in fish feed is crucial, since they are not able to be synthesized due to the lack of the enzyme L-gulonolactone oxidase, which is necessary to convert L-gulonic acid into vitamin C. And this is very crucial vitamin, by the way, for the anti-stress formulas sold in the Egyptian environment. What is the physiological role of vitamin C? The physiological role of vitamin C is related to stress, is based on the steroidogenesis, inhibition through the peroxidation of polyunsaturated lipids, and enhancement of the immune system. However, the effect of this supplement on the cortisol biosynthesis, as we all know that the corticosteroid of fish is cortisol, it's unsaturated corticosterols. So the cortisol biosynthesis could not be demonstrated in fish. Researchers have reported the beneficial immunomodulatory and antioxidant effects of vitamin C in stressed fish, stating that dietary ascorbic acid supplement alleviate chronic stress effects, which is great. Another player is lipids and fatty acids. The importance of lipids in stress response is based on the formation of eicosanoids, particularly prostaglandins, either in fish or maybe in large animals or maybe in mammals and so on. They are all similar. Concretely, the arachidonic acid, unsaturated essential fatty acids, to eicosanoids, acting as endocrine, paracrine, and autocrine modulator of secretory mechanisms in various organs. Mainly due to its key nutritional role, other fatty acids like docosahexaenoic and eicopantenoic acids, DHA and EPA or EPA, have been studied. Similarly, it has stated that several HUFAs, highly unsaturated fatty acids, for instance ABA, are also eicosanoids precursor or prostaglandin precursor. Besides, eicosanoids, more fundamental processes like alterations in membrane properties, cellular signal transduction, and supported to contribute to the consistent effect of dietary DHA and EPA on growth, stress resistance, and certain immune responses. Another new and recent emerging player in the market of anti-stress formulas and stress alleviation formulas in the Egyptian market is a nucleotides. The roles of nucleotides and the metabolites in fish diets have been studied for almost 20 years, and most research has shown rather consistent and encouraging beneficial results in health management of both marine and freshwater fish. Researchers have reviewed the influence of dietary nucleotides or innate and adaptive immunity in fish and suggested that dietary nucleotides would support lymphoid tissues that have limited de novo synthesizing capacity, which is great immunostimulant effect, great humoral stimulating effect, great cellular stimulating effect. Thus, nucleotides is a recent emerging player in the Egyptian market. To this moment, I have reached the end of my lecture. I really appreciate the kind invitation which I have received from Professor Muhammad Usman, and I would like to thank all of you for attendance and for the active participation in this very important session. Thank you very much and have a great day.